How do you make antibodies?
Well, there are several ways to make antibodies. And first, you have to identify antibodies to cancers. And you could do hybridoma technology. So that means you use mice or rabbit to raise antibodies against the tumor antigens. So you basically immunize mice or rabbit with the tumor antigens and let the mouse and the rabbit to make antibodies for you. And the other approach we are more commonly used now is actually use the phage display. So use the bacterial virus, and we have a bacterial virus library. So the phage display the antibodies on their surface, and we use that library to screen the antibody to the target. Once we find the antibody, we have to make a large amount to the animal testing and other functional biochemical and cell biology test. To do that, we need to grow the antibodies, and basically you clone the antibody into the cells, into like mouse cells or human cells, and then you grow the cells, and you collect the supernatant from the cells, and purify use the like protein A columns, so affinity columns, to actually purify the antibodies from the supernatant of cells. And then you quantitate it and dilute it enough amount and inject into the mice, or just do that in the cells, cell assays.
Then how do you get cancer cells, the tumor? Do you, in the lab environment, do you need to inject antibody into a tumor?
Yeah. So we have animal facility. So we grow the tumors in mice, and then we inject antibody to treat the mice, just like treat our patients. And you see the mice tumor shrink. You know it actually antibody works.
Okay, and
how animal get tumor at the first place?
Oh yeah, animal, yeah. So the mice is like a typical mice. It's a mice could grow the human tumors. So the mice we use is immunocompromised mice. So they don't reject the human tissues. So they could actually grow the human tumors in mice. So that's the model we could use. It's a still human cancer model, but in mice.
Oh, what are cancer cells?
It's cancer cells, like liver cancer cells, right?
Oh, what are they?
Yeah, cancer cells is just like a tumor. So tumor are actually it's just different from normal cells, and they are not healthy, and that's how they cause tumors. So the cancer cells are the cells abnormally grown. Unlike the normal healthy cells, they don't grow that fast.
And how quickly since you guys are doing liver cancer, like how quickly does one liver cancer cell go through the process of mitosis and like duplicate?
Well, normal cells do the duplicate in the normal like 24 or 26 hours, on the tissues, and the cancer cell, they basically just grow much faster. And that's not only the growth rate. Cancer cell could grow to disrupt the so-called contact inhibition. So the normal healthy cells, they cannot grow over each other, okay? So the cancer cells can grow over each other because they don't have the boundary for the cell contact. So the normal healthy cells, when they grow, when they touch each other, they will basically stop growing. But the cancer cell, they will still step on each other. So that's why they are more aggressive.
How quickly do they Like how quickly do liver cancer cells?
It depends. It depends. Different liver cancer cells also grow different rate. And the major problem is they just don't stop. They just keep growing. And when normal healthy liver cells, for example, they will be more restrained, and they won't grow unlimited. They will stop at some time. So the cells like normal healthy cells, the reason they will actually when they communicate each other, they know where's the boundary, and they usually don't grow over the boundary. And that's how it's very complicated, because in biology we still don't quite understand, for example, what the size of our organ, for example, is determined by what kind of mechanism. How cells know that's the end and you don't grow anymore, right? And how this organ grows such a particular size and then it does not grow anymore. You know, even like human, when you grow like this height, why you cannot grow forever? You know, those things is actually related to the genetics, biochemistry, and essentially normal healthy cells, they know the limit. Okay, so that's how they regulate it. But the cancer cells, they basically irregulate it, so they don't know the limit. They sometimes just overgrow, and that's how cause the problem.
Okay.
How antibody treatment of cancer is different from chemo treatment?
Well, it's just different. Yeah, antibody treatment is also kind of chemo treatment.
Okay.
It's just a new type of chemo treatment, because traditional chemotherapy, they don't particularly distinguish the tumor cells from normal cells, because they only target the fast-growing cells. But the tumor cells are fast-growing cells, so they still could target more tumor cells than normal cells, but they're just not as specific as antibody therapy, because antibody particularly target only cancer cells. And for chemotherapy, for example, you have some side effect. When people treat with traditional chemotherapy, you lost hair, you have a lot of toxicity, because they also destroy some normal cells, normal healthy cells. And that's not the case for antibody therapy, because they are more specific. They are not supposed to actually target a lot of other normal cells. But does not mean antibody therapy is 100% safe. And they also have some other side effect, but just not the same and not as much side effect as traditional chemotherapy we usually see. And in general, antibody therapy is less toxic and has less side effect. And the patient treated with antibody therapy has a better life quality, because the patient treated with antibody therapy, they basically still could walk, and they still could do a lot of things. And when chemotherapy may destroy a lot of patients' life quality.
Thank you for this.
Sure. Yeah.
Thank you.